Hey guys, welcome to part two of our episode on glycogen metabolism. Um, if you haven't watched part one, I would suggest you do because it's going to make sense since we're starting in basically the middle now. Okay, if you're unfamiliar with the structure of the podcast, AI and I have a talk about topics that I'm unfamiliar with, and yeah, AI explains the topics. I listen and I try to learn, and perhaps you can learn something throughout this process too. So, Grog, let's talk about glycogen synthesis. We talked about glycogen breakdown in part one. Let's talk now about glycogen synthesis, glycogenesis.
All right. Glycogenesis is the process where the liver and muscle cells build glycogen from glucose to store energy, mainly triggered by insulin when blood glucose is high. Glucose is converted to glucose 6 phosphate, then to glucose 1 phosphate, which is activated into UDP glucose by enzymes like UDP glucose pyrophosphorylase. And glycogen synthase adds these units to glycogen synthase via alpha 1 4 bonds, with branching enzyme creating alpha 1 6 bonds for a branched structure. This stores excess glucose in the cytoplasm as glycogen granules for later use, like during fasting or exercise. Want to dive into the specific enzymes or how it balances with glycogenolysis?
So, what is UDP glucose and how exactly is glycogen formed from UDP? It's the UDP part that's confusing me.
UDP glucose is a high energy molecule formed in the cytoplasm when glucose 1 phosphate reacts with UTP, uridine triphosphate, via the enzyme UDP glucose pyrophosphorylase, acting as the activated glucose unit for glycogen synthesis. Glycogen synthase takes UDP glucose and adds the glucose to the non-reducing end of a glycogen chain, forming an alpha 1,4 glycosidic bond, releasing UDP in the process. The branching enzyme later adds alpha 1,6 bonds to create glycogen's branched structure. The UDP, uridine diphosphate, is just a nucleotide carrier that activates glucose, making it ready to be linked into glycogen in liver and muscle cells. Want more on the enzyme steps or why UDP is key?
Yeah, I want to understand the uridine part. Then I want us to start talking a bit more about how glucose will become G6P, then G1P, and eventually UDP before it becomes glycogen. So we have all these steps. I wonder why they're in this order. We get ATP in the first step and use it for glucose to become G6P. Correct me if I'm wrong. And we also need hexokinase. Why specifically hexokinase or glucokinase in the liver? And why from G6P to G1P we need phosphoglucomutase? And what is UTP? Why do we need it for the next step?
Uridine in UDP glucose is a nucleotide base that, when paired with two phosphate groups, UDP, acts as a carrier to activate glucose 1 phosphate into UDP glucose, providing the energy and structure needed for glycogen synthase to add glucose to glycogen chains in the cytoplasm of liver and muscle cells. The process starts with glucose being phosphorylazed to glucose 6 phosphate, G6P, by hexokinase in muscles or glucokinase in the liver, using ATP because glucokinase in the liver has a lower affinity, suited for high glucose levels after meals. G6P is then converted to glucose 1 phosphate, G1P, by phosphoglucomutase, which shifts the phosphate to prepare it for activation, as G6P can't directly form glycogen. G1P reacts with uridine triphosphate, UTP, a high-energy molecule like ATP, via UDP glucose pyrophosphorylase to form UDP glucose, with UTP providing the energy needed to link glucose into glycogen. And the steps are ordered to efficiently activate and position glucose for polymerase action while maintaining metabolic control. Want more on specific enzymes or the energy dynamics of these steps?
Yeah. So okay. we have uridine. Does uridine have to do anything with urine? And then you have glycogen synthase. What does glycogen synthase do? So it adds a glucose basically to the glycogen. And then, so we use UTP and why is UTP energetically equivalent to ATP consumption? And then basically we get UDP out.
Uridine in UDP glucose has nothing to do with urine. It's a nucleotide base, like adenine in ATP, that forms part of UDP, which activates glucose for glycogen synthesis in the cytoplasm. Glycogen synthase takes UDP glucose and adds the glucose unit to the non-reducing end of a glycogen chain, forming an alpha-1,4 glycosidic bond, releasing UDP. UTP is energetically equivalent to ATP because both are high-energy triphosphates, providing similar energy to drive the reaction. The process converts glucose 1-phosphate to UDP glucose using UTP, and after glycogen synthase acts, UDP is released and recycled back to UTP, ensuring efficient energy use for glycogenesis. Want more on the synthase mechanism or UTP's role?
So uridine is like uracil.
Yeah, uridine is a nucleoside that contains uracil as its base, linked to a ribose sugar, and in UDP glucose, it's part of UDP, uridine diphosphate, that activates glucose for glycogen synthesis. Uracil is the nitrogenous base itself, found in RNA, while uridine is the whole package, base plus sugar, used in metabolic processes like glycogenesis in the liver and muscles. The distinction matters because uridine's role in UDP provides the energy.
Keep going. Why'd you stop?
All right, so we were talking about uridine being a nucleoside with uracil as its base, linked to ribose sugar, and in UDP glucose, it's key for activating glucose during glycogenesis in liver and muscle cells. This UDP glucose is used by glycogen synthesis to add glucose to glycogen chains, forming alpha-1,4 bonds, while UTP, similar in energy to ATP, drives this process by providing the needed energy, releasing UDP afterward. These steps ensure glucose is efficiently stored as glycogen, with uridine's role in UDP making it possible to link glucose units without relying on ATP directly.
So we have UDP, we have glycogen synthase, and then we end up with, we have UDP glucose, and then glycogen. Then with glycogen synthase as help, we end up with glycogen with an extra glucose and uridine in the side.
Yeah, exactly. UDP glucose is used by glycogen synthesis in the cytoplasm of liver or muscle cells to add a glucose unit to a glycogen chain, forming an alpha-1,4 glycogen bond, and releasing UDP, uridine diphosphate, as a byproduct. The uridine in UDP is just part of the nucleotide carrier that activates glucose, and after the reaction, UDP is recycled back to UTP for more rounds of glycogenesis. This process builds glycogen stores for later energy use, regulated by insulin. Want to dive into the branching process or UDP recycling?
So how come glycogen synthase adds glucose units in an alpha-1,4 and not in an alpha-1,6? How do we add things in alpha-1,6?
Glycogen synthesis adds glucose units from UDP glucose to form alpha-1,4 glycogen bonds because its active site is specific to creating linear chains by linking the carbon-1 of one glucose to the carbon-4 of another, extending glycogen in the liver and muscle cytoplasm. Alpha 1,6 bonds are added by a separate enzyme, the branching enzyme, amylo 1,4 to 1,6 transglucosidase, which takes a segment of about 7 glucose units from a chain's non-reducing end and attaches it to a carbon 6 elsewhere, creating a branch point for compact storage. This division of labor ensures glycogen's linear growth via synthesis and branched structure via the branching enzyme for efficient storage and breakdown. Want more on the branching enzyme or regulation of these bonds?
Well, in a sentence, so how do we add alpha 1,6? Up to now, we've only talked about how we add the alpha 1,4 linkage.
Alpha 1,6 glycogen bonds are added by the branching enzyme, which transfers a short chain of glucose units from a linear alpha 1,4 linked glycogen chain to the carbon 6 of another glucose unit in the liver or muscle cytoplasm,
I see.
creating...
So, yeah, we talked about this in the part 1, I just remembered. Okay, so glycogen synthesis, how does it start? So, because glycogen synthase adds only to pre-existing chains that has 4 glycosyl residues. Tell me about the priming function, and tell me about the protein glycogenin.
Glycogen synthesis starts with glycogenin, a protein in the liver and muscle cytoplasm that acts as a primer by attaching a glucose unit from UDP glucose to itself, forming a short chain of about 4-8 glucose residues linked by alpha 1,4 bonds. This initial chain serves as the foundation for glycogen synthesis to add more glucose units from UDP glucose, extending the glycogen molecule. Glycogenin's priming is crucial because glycogen synthesis can't start a chain from scratch and needs this pre-existing glucose stub to kick off glycogenesis.
So glycogenin, this priming function starts things off.
Yeah, glycogenin kicks off glycogenesis by attaching UDP glucose to itself in the liver or muscle cytoplasm, creating a short alpha 1,4 linked glycogen chain that acts as a primer for glycogen synthesis to build onto. Without glycogenin, glycogen synthesis can't start a new glycogen molecule from scratch, as it only adds to existing chains. This priming ensures efficient glycogen storage for later energy use. Want more on glycogenin's role or how the chain grows?
Well, how does tyrosine play a role in this?
Tyrosine plays an indirect role in glycogenesis as a glucogenic amino acid that can be broken down in the liver to produce oxaloacetate or fumarate, which feed into gluconeogenesis to generate glucose 6-phosphate for glycogen synthesis. Specifically, tyrosine is metabolized in the cytoplasm and mitochondria to fumarate, which enters the Krebs cycle and can be converted to glucose precursors, supporting glycogen production when glucose is scarce. Its role is secondary, as glycogenesis primarily uses dietary glucose, but tyrosine contributes under fasting conditions to maintain glycogen stores. Want more on tyrosine metabolism?
UDP glucose donates the first glucosyl residue, attaching it to tyrosine in the glycogenin. And then this glycogenin extends the glucose chain by up to 7 additional residues.
Yeah, exactly. UDP glucose donates the first glucose to a tyrosine residue on glycogenin in the liver or muscle cytoplasm, where glycogenin's catalytic site links it via an alpha 1,4 glycogen bond, then adds up to 7-8 more glucose units from UDP glucose to form a short primer chain. This chain, still attached to glycogenin, gives glycogen synthesis a starting point to extend the glycogen molecule with more alpha 1,4 linked glucose units. The process sets the foundation for efficient glycogen storage, regulated by insulin. Want more on how glycogen hands off to synthesis or something else?
And so this branching enzyme, it transfers up to a block of 7 residues from a growing chain and creates a new alpha 1,6 linkage.
Yes, the branching enzyme amylo 1,4 to 1,6 transglucosidase takes a block of about 6 to 8 glucose residues from the non-reducing end of a growing glycogen chain and transfers it to a glucose unit elsewhere on the chain, forming a new alpha-1,6 glycogen linkage to create a branch point in the liver or muscle cytoplasm. This branching allows glycogen to form a compact, tree-like structure, increasing storage efficiency and providing multiple sites for glycogen synthesis.
So where does this happen? Wait a sec. So when does the branching enzyme act? Because last time with the branching enzyme, we were saying that it removes stuff, so there's one glucose left and we can attack that glucose to remove it.
The branching enzyme acts during glycogenesis in the liver or muscle cytoplasm, taking a block of about 6 to 8 glucose residues from a glycogen chain's non-reducing end and attaching it via an alpha-1,6 glycogen bond to create a new branch, which happens after glycogen synthesis extends linear chains. In glycogen breakdown, glycogenolysis, discussed last time, the same enzyme, as part of the debranching process, transfers 3 glucose units from a branch to another chain, leaving 1 glucose at the alpha-1,6 branch point, which alpha-1,6 glycogenin then removes. The branching enzyme's role in synthesis builds the glycogen tree, while in breakdown, it helps dismantle branches for glucose release. Want more on when it kicks
So
in?
it's a different enzyme. One is a branching enzyme, the other is a debranching enzyme. Can we compare their mechanism of action a bit, these two enzymes?
The branching enzyme, amylo-1,4 to 1,6 transglucosidase, and debranching enzyme work in the liver and muscle cytoplasm, but have opposite roles in glycogen metabolism. During glycogenesis, the branching enzyme takes a block of 6 to 8 glucose units from a glycogen chain's non-reducing end and attaches it to a glucose unit elsewhere via an alpha-1,6 glycogenin bond, creating a new branch for compact storage. In glycogenolysis, the debranching enzyme's transferase activity moves 3 glucose units from an alpha-1,6 branch point to another chain's non-reducing end, forming an alpha-1,4 bond, and its alpha-1,6 glycogenin activity removes the remaining single glucose, freeing it and allowing phosphorylase to continue breaking down the chain.
Question. So with glycogen, it's a polar molecule. It relates with water. What are the effects of this relation? How does it affect function of things?
Glycogen, a polar polysaccharide, due to its many hydroxyl groups, interacts strongly with water in the cytoplasm of liver and muscle cells, forming a hydrated, granular structure that enhances its storage efficiency and accessibility. This polarity allows glycogen to be easily surrounded by water, keeping it soluble and readily available for enzymes like glycogen phosphorylase or synthesis to act quickly during glycogenolysis or glycogenesis. However, its polarity means it can't cross the hydrophobic cell membrane, so it stays trapped in the cytoplasm, ensuring localized glucose storage and release for energy or blood sugar
So in
regulation.
a sentence, nice and simply,
Glycogen's polarity lets it mix with water in the cytoplasm, making it easy for liver and muscle cells to store and break it down for energy or blood sugar, but it stays trapped inside cells. Want to dive deeper into its storage or move to another topic?
what do you mean it stays trapped? Why would it stay trapped?
Glycogen stays trapped in the cytoplasm of liver and muscle cells because, as a large, polar molecule with many hydroxyl groups, it can't cross the hydrophobic lipid bilayer of the cell membrane without specific transporters, which don't exist for glycogen.
Okay,
This keeps
fair enough. Okay, let's talk a bit about regulation of glycogen metabolism. So we've talked when we have glycogen mobilization regarding to when we eat. And we need glycogen synthase to replenish liver glycogen stores. So I think we're good with that actually. So let's talk about allosteric regulation of phosphorylase.
allosteric regulation of glycogen phosphorylase in liver and muscle cells controls glycogen breakdown by responding to metabolic signals. In muscles, glucose 6-phosphate and ATP bind to phosphorylase, inhibiting it when energy is high, while AMP activates it during energy demand, like exercise. In the liver, glucose acts as an allosteric inhibitor, slowing glycogenolysis when blood sugar is sufficient, ensuring glycogen is broken down only when needed. Want more on how this ties to hormonal regulation or specific triggers?
So tell me more about the allosteric regulation. So we have glycogen phosphorylase, and AMP and ATP and G6P, they touch it. So let's start with AMP. So AMP, what is it first of all? How does it relate to ATP? And tell me how it activates phosphorylase.
AMP, adenosine monophosphate, is a low-energy molecule related to ATP, adenosine triphosphate, formed when ATP loses two phosphate groups, signaling low cellular energy. In muscles, AMP binds to an allosteric site on glycogen phosphorylase, changing its shape to boost its activity, speeding up glycogenolysis to release glucose 1-phosphate for ATP production during exercise. ATP and glucose 6-phosphate, G6P, when abundant, bind to different allosteric sites, inhibiting phosphorylase to slow glycogen breakdown when energy is high.
And what in the structure of AMP makes it ideal for this job?
AMP's structure, with an adenine base, ribose sugar, and a single phosphate group, makes it ideal for allosteric activation of glycogen phosphorylase because its low energy state signals energy depletion, and its shape fits a specific regulatory site on the enzyme. This binding shifts phosphorylase to an active form, speeding glycogenolysis to produce glucose 1-phosphate for ATP in muscle cells during energy demand. Unlike ATP's 3 phosphates, AMP's single phosphate allows distinct binding, ensuring the enzyme responds to low energy cues. Want more on the binding site or liver-specific regulation?
And then ATP and glucose 6-phosphate, instead of, they kind of have the opposite effect. So they inhibit phosphorylase. So inhibiting phosphorylase, slowing the glycogen breakdown. Interesting. So the opposite effect. Okay, and let's talk about the allosteric regulation of glycogen synthase a bit now. So we have glycogen synthase, and it's allosterically activated by G6P again. So this means that G6P not only slows down the breakdown of glycogen, but it also increases the pace where glycogen is built, right?
Exactly. Glucose 6-phosphate, G6P, allosterically activates glycogen synthase in the liver and muscle cytoplasm by binding to it, shifting it to an active form that speeds up glycogenesis, adding glucose to glycogen chains. At the same time, G6P inhibits glycogen phosphorylase, slowing glycogen breakdown, so it promotes storage when glucose levels are high. This dual role ensures efficient glucose storage and prevents futile cycling between glycogen synthesis and breakdown. Want more on how G6P's binding works or its hormonal regulation?
Okay, so let's talk a bit about in the liver and in the muscle. How is that allosteric control the same?
In both liver and muscle, allosteric control of glycogen metabolism involves glucose 6-phosphate, G6P, activating glycogen synthase to promote glycogenesis and inhibiting glycogen phosphorylase to slow glycogenolysis, ensuring glucose storage when energy is abundant. However, in muscles, AMP activates phosphorylase during low energy, like exercise, while in the liver, glucose itself inhibits phosphorylase to regulate blood sugar. These shared and tissue-specific controls balance glycogen synthesis and breakdown based on energy needs.
Tell me this last thing again, you said? So you said that they have the similarities. What's the difference you said in the end?
In liver and muscle, allosteric control is similar. Glucose 6-phosphate, G6P, activates glycogen synthase and inhibits glycogen phosphorylase to favor glycogen storage. The key difference is that in muscles, AMP activates phosphorylase to boost glycogen breakdown during exercise, while in the liver, glucose directly inhibits phosphorylase to regulate blood sugar levels. This
I don't
reflects muscle.
understand this difference.
All right, let's simplify the difference in allosteric control between liver and muscle for glycogen metabolism. In both, glucose 6-phosphate, G6P, promotes glycogen storage by activating glycogen synthase, builds glycogen, and inhibiting glycogen phosphorylase, breaks glycogen. The difference is, in muscles, AMP, low energy signal, activates phosphorylase to break down glycogen for energy during exercise, while in the liver, glucose itself shuts down phosphorylase to prevent glycogen breakdown when blood sugar is already high.
Okay, let's talk about regulation of glycogen metabolism by covalent modification. So tell me in a sentence what covalent modification is and how it's mediated by the addition of a phosphate group. Also, talk to me about addition of a phosphate group, why we call it phosphorylation. Phosphorylation, I thought, was breaking down the glycogen. And why is it catalyzed by protein kinases?
Covalent modification, specifically phosphorylation, is the addition of a phosphate group to enzymes like glycogen phosphorylase or synthase, altering their activity to regulate glycogen metabolism in liver and muscle cells. Phosphorylation, named for adding a phosphate group from ATP, activates glycogen phosphorylase to break down glycogen into glucose 1-phosphate and then activates glycogen synthase to halt glycogen synthesis, with protein kinases catalyzing this by transferring the phosphate due to their specificity and energy-driven mechanism. This process, triggered by hormones like glucagon or epinephrine, ensures rapid glycogen breakdown during low blood sugar or exercise. Want more on specific kinases or how this differs from allosteric control?
Tell me about how this is a reversible modification and we can remove phosphate groups through dephosphorylation, and how we can catalyze them with protein phosphatases. Also, sorry guys, I'm a bit in a rush because I want to keep them to 30 minutes. Tell me about phosphorylase, how it's activated by CAMP-dependent phosphorylation. What does this mean? Talk to me about the CAMP cascade in general, how it results in phosphorylation of a hydroxyl group. Tell me about serine residues and and this active-inactive state. And let's talk a bit about the phosphorylated enzyme, like how it's less sensitive to allosteric inhibitors. Yeah.
All right, let's keep this tight since you're aiming for 30 minutes. Reversible covalent modification in glycogen metabolism involves phosphorylation, adding phosphate groups via protein kinases, and dephosphorylation, removing them via protein phosphatases to toggle enzymes like glycogen phosphorylase and synthase between active and inactive states. Phosphorylation, catalyzed by protein kinases like protein kinase A, PKA, adds a phosphate from ATP to serine residues on phosphorylase, activating it to break down glycogen. Dephosphorylation by protein phosphatases, like PP1, removes the phosphate, inactivating phosphorylase and activating glycogen synthase for glycogen synthesis. The CAMP cascade starts when glucagon or epinephrine binds liver or muscle cell receptors, boosting CAMP, which activates PKA to phosphorylate hydroxyl groups on serine residues, making phosphorylase more active and less sensitive to allosteric inhibitors like glucose or ATP, ensuring rapid glycogen breakdown during low blood sugar or exercise.
Okay, talk to me 2 sentences about phosphorylase A and B, activated versus less active, about protein kinase and protein phosphatase and PI, and about induction of CAMP cascade, how it has an opposite effect on glycogen synthase. Talk to me a bit about phosphorylation of glycogen synthase. Talk to me a bit about glycogen synthase A and B. 3 sentences total.
Phosphorylase A is the phosphoracided active form of glycogen phosphorylase, driven by protein kinase A, PKA, via the CAMP cascade triggered by glucagon or epinephrine, while phosphorylase B, The dephosphorylated, less active form is restored by protein phosphatase 1, PP1, which removes the phosphate in liver and muscle cells. The CAMP cascade activates pKa to phosphorylate glycogen synthase, converting it to the inactive synthase B form, halting glycogen synthesis, while dephosphorylation by PP1 produces active synthase A, promoting glycogen production.
Got it.
This
Perfect. Thank you so much, guys, for watching, and peace.